In 2023, a 5-year-old boy named Terry died, just weeks after receiving an experimental $800,000 gene therapy. His death, tragically, wasn't an isolated incident, but a stark, horrifying window into a hidden crisis in medical innovation. This isn't just about the eye-watering price tag of these new treatments. It's about the true devastating costs that companies often don't want you to see, the undisclosed risks, the rushed timelines, and the profound ethical dilemmas quietly unfolding in labs right now. We're talking about treatments with incredible promise, but also a dark underbelly. We're going to dig into Terry's story, uncover how these therapies are actually developed, and reveal why patients and their families are often kept in the dark about crucial data. And stick with me, because the last part of this is the piece almost everyone gets wrong about these multi-million-dollar medical breakthroughs. And it's the part that determines who truly pays the ultimate price. The shocking reality is that despite life-changing potential, gene therapies carry a terrifying and often unspoken price beyond the sticker shock. Just last month, the medical community grappled with the tragic death of an 18-month-old child treated with an $800,000 gene therapy, prompting the FDA to issue a partial clinical hold on future trials. This isn't an isolated incident. It's a stark reminder of the immense risks inherent in a burgeoning industry that promises miracles but sometimes delivers heartbreak. We're talking about treatments that can cost more than a house, yet come with potentially fatal side effects that aren't fully understood until it's too late. The company behind this specific therapy, Astellas Pharma, saw its stock dip slightly, but the human cost for the family is immeasurable. This case isn't just about a single adverse event. It highlights a critical blind spot in how we evaluate, commercialize, and ultimately pay for cutting-edge medical innovation. We need to look beyond the dazzling scientific breakthroughs and confront the brutal complexities. The $800,000 one-time gene therapy isn't a singular payment. It's often merely a down payment on a lifetime of additional, often unadvertised, costs. The initial allure of a one-time permanent cure for a staggering sum, such as the widely reported $2.1 million for Zolgensma, onisimagene abiparvovec, for spinal muscular atrophy, or $850,000 for Luxturna, voretigene neparvovec, for inherited retinal dystrophy, often fails to include the full financial and medical picture. What isn't always factored into that initial sticker price are the critical long-term monitoring regimens, specialized follow-up diagnostic tests like PCR assays for viral vector persistence, or even potential booster doses if the initial gene expression wanes, all of which can add hundreds of thousands more to the total. Consider early gene therapies for severe combined immunodeficiency, SCID, like those developed for X-linked SCID. While groundbreaking, many required subsequent treatments, such as bone marrow transplants, because the initial therapy either failed to provide sustained benefit, sometimes due to neutralizing antibodies, or presented severe side effects like insertional mutagenesis. Dr. Jeffrey Leiden, former CEO of Vertex Pharmaceuticals, aptly noted that the industry is still learning the true long-term efficacy and safety profiles of these novel treatments. Patients receiving these therapies also need regular checks for immune responses to the viral vector, potential off-target effects on non-target cells, and to ensure the treatment's efficacy is sustained over many years, sometimes through yearly lumbar punctures for CNS-targeting therapies. These aren't just medical necessities. They're ongoing financial and time burdens on patients and their families, often requiring specialized clinics hundreds of miles away. It's a critical oversight when we discuss the true affordability of these pioneering treatments. The next crucial question we must ask is, beyond the financial implications, what happens inside the body when these cutting-edge therapies encounter an unexpected or adverse biological response? But even if the financial costs are just the beginning, there's another, more insidious danger. Your own immune system can turn into a hidden time bomb. The core mechanism of most gene therapies is to use a viral vector, often a modified adeno-associated virus, or AAV, to deliver new, corrective genetic material into a patient's cells. Think of it like a microscopic delivery truck. But what happens when your body sees that truck as an invasion? In 1999, the world learned this lesson in the most tragic way with the death of Jesse Gelsinger, a young man treated for a rare metabolic disorder. His body mounted an overwhelming immune response to the adenovirus vector used in his therapy, leading to multi-organ failure. It was a stark reminder that while these modified viruses are designed to be harmless, your immune system doesn't always distinguish between a therapeutic delivery vehicle and a genuine pathogen. This isn't just an immediate risk, it's a long-term threat. An unpredictable immune response can manifest years after the initial treatment, leading to chronic inflammation, organ damage, or the outright rejection of the very cells the therapy was meant to fix. For example, a significant portion of the population, an estimated 20% to 40%, already has pre-existing immunity to common AV variants due to prior exposure to wild viruses. This can render the gene therapy ineffective or even dangerously provoke an immune cascade, as the body's defenses are already primed to attack the viral vector. It's a fundamental biological hurdle that isn't always accounted for in the one-time promise. And what happens if the gene isn't delivered precisely where it's supposed to go, or if it influences other genes in unintended ways? But what happens when that delivery truck, the viral vector, doesn't just deliver its package, but then crashes into your body's vital infrastructure? Even if the gene therapy gets to the right cells, it can still go rogue, leading to devastating off-target effects. This is where the therapy mistakenly inserts into unintended parts of your genome or activates other, unwanted genes. The most terrifying of these is insertional mutagenesis. Imagine the viral vector integrating itself right into a tumor suppressor gene, effectively disabling it, or worse, activating an oncogene, which could potentially lead to cancer. We've seen this play out in real time. In the early 2000s, trials for SIDX-1, a gene therapy for bubble boy disease, tragically saw 5 out of 20 children develop leukemia, with one child dying, because the corrective gene inserted near a cancer-promoting gene. Even with advancements in gene editing like CRISPR, predicting and preventing these off-target integrations remains a monumental challenge. Dr. Fyodor Urnov, a gene editing pioneer at the Innovative Genomics Institute, has publicly stated that while CRISPR is more precise, no editing tool is 100% specific. The long-term consequences of these subtle, often unpredictable, genomic changes are still being debated and uncovered. What if the therapy affects cell lines beyond the immediate target, subtly altering various systems in your body over decades, leading to unforeseen systemic issues? The more we push the boundaries, the more we confront the invisible complexities of our own biology. This brings us to a question that chills many scientists: the unseen inheritance, altering future generations? But what if these groundbreaking modifications don't just affect the patient, but quietly rewrite the genetic code of future generations? The critical, often understated, risk of gene therapy isn't solely about the immediate recipient, but the silent possibility of germline transmission, where the therapeutic genetic material could inadvertently integrate into a patient's sperm or egg cells. Should this occur, the gene therapy's effects, both intended therapeutic benefits and unforeseen off-target mutations, could be passed down to offspring, fundamentally altering the human gene pool without the consent of those future individuals. This isn't a speculative scenario from a sci-fi novel. It's a real, albeit low-frequency, concern for treatments specifically designed to target somatic cells. Consider the 2017 study led by Drive Susan Hall in Human Gene Therapy, which meticulously documented unexpected germline transfer of adeno-associated virus, AAV, vectors in several animal models. While direct, incontrovertible human evidence remains limited and controversial, the ethical and societal implications of accidental germline editing are profound. It ventures into uncharted territory for human reproduction and evolution, raising complex questions that extend far beyond individual patient consent forms. The current stringent regulatory environment, spearheaded by agencies like the FDA in the United States and the European Medicines Agency, EMA, strictly prohibits intentional germline editing in humans precisely due to these profound concerns. However, The potential for accidental, low-frequency transmission remains a silent but persistent shadow in the development of somatic cell gene therapies, a risk that's often overshadowed by the immediate, life-altering promise of a cure for debilitating conditions. What happens, then, when the cure itself, meant to be a one-time fix for an individual, takes on a life of its own, evolving beyond our current understanding and control? But what happens when the cure itself doesn't just treat, but evolves, spreading beyond the patient in ways we can't control? This is the ultimate, most profound long-term risk: the potential for gene therapy vectors or modified cells to spread. Imagine a scenario where the very delivery truck we discussed, the viral vector, doesn't just deliver its package and dissipate, but somehow replicates, or is shed, and inadvertently transfers those altered genes to others, or even to the environment. Scientists like Dr. Kevin Isvelt at MIT have popularized the concept of gene drives, a synthetic biological mechanism designed to rapidly spread a modified gene through a population. Initially envisioned for noble goals like malaria eradication by altering mosquito populations, the implications for human applications are chilling. The unspoken fear is that an engineered virus or modified cell, perhaps designed to correct a specific genetic defect, could become infectious. What if a patient, after receiving a gene therapy, inadvertently sheds these modified viruses? Dr. Francis Collins, former director of the NIH, has often emphasized the stringent containment measures in labs, but the real world is far less controlled. Consider a situation where a patient's altered cells become viable outside their body, perhaps integrating into environmental bacteria or even jumping to other humans, initiating a cascade of unintended genetic modifications across a population. The biotech industry, understandably, often downplays these worst-case evolutionary scenarios, prioritizing immediate efficacy. They focus on the $3.5 million price tag for a single dose of Hemgenix, not the distant future of biological containment failure. This is the true, unspoken cost and danger. A future where the remedies we create could become a self-propagating, uncontrollable force, rewriting the very blueprint of life in ways we can't predict, let alone recall. So, while the promise of a one-time cure for $800,000 might sound revolutionary, what we've unpacked today reveals a far more complex and potentially perilous reality. The rush to deliver these high-stakes treatments, often without decades of long-term data, means we're not just treating individual patients. We are, in a very real sense, altering the fundamental building blocks of life. This isn't just about managing side effects. It's about confronting the evolving, long-term implications for individuals, for future generations, and even for the broader biological landscape. The narrative often centers on immediate breakthroughs, but it's crucial to push for transparency, robust monitoring protocols, and a more honest societal conversation about the true scope of biological engineering we are embarking on. Considering these profound, long-term risks, do you believe the current regulatory frameworks for gene therapies are adequate, or do they need a fundamental reevaluation to address these unseen possibilities? Let me know your thoughts in the comments below. If you're interested in understanding the hidden mechanisms behind today's biggest tech and biotech stories, hit that subscribe button. And if you want to dive deeper into how these cutting-edge therapies are priced and what that means for our healthcare systems, watch my breakdown on why those one-time cures sometimes cost more than a house.